Okay, let's look at an easy way to extract DNA from strawberries. We use strawberries in this experiment because they have 8 pairs of chromosomes. Humans only have 2 pairs of our chromosomes. We have 46 chromosomes, 2 pairs of all the 23 that we get from each parent. But these guys here have 8 pairs. They've got tons and tons of DNA. So there's relatively large amounts to extract. So the materials that we need a strawberry, plastic bag, and a cup of salt, some detergent, I use Palmolive, and a couple of plastic cups, one with a coffee filter over the top, some distilled water, and also some ice cold rubbing alcohol, preferably 91%, the good stuff. Okay, so here's what we do first. Take your strawberry and make sure you pick off all the green stuff, and then put it into a plastic bag. And get someone to start mushing this up. We want to break open the cells, start the process of extraction, and in the meantime, while someone is mushing this up, we want to get it to a nice paste consistency. We want it to be completely smushed. While someone is doing that, someone else can begin preparing the extraction buffer. So to make that, we'll take a cup and fill it about half full with distilled water. And then we're going to add a scoop of salt and 2 good squirts of detergent. Now the purpose for this is to give the DNA the right environment when it is removed from the cells so that it doesn't just turn into a big tangled mess. The purpose of the salt is to help create the correct ionic environment to stabilize the DNA molecules when they come out. And the detergent is to help break open cell membranes, because cell membranes are similar in structure to detergent and soap molecules. They are able to mix together, and that helps to dissolve or solubilize the cell membranes. Okay, so I'm going to mix this up. You don't want bubbles, so mix this very, very gently. And in the meantime, someone else has been smushing up your strawberries. Let's see how that's going. Okay, so I've been smushing this for a while. I've been making sure it's all nice and mashed. I've squeezed it all at the bottom of the bag so that when I open up the top, it doesn't all come out. And now I'm going to take about half of my buffer solution here and pour it in. Only need about half. And then close this up. And when you close this up, you want to try to keep out the air as much as possible. So I'm just going to lay it flat when I close it so that not much air remains. Push out the air bubbles. And now what we're going to do is we're going to continue to smush the strawberries. We're going to mix this around. But while you do that, you want to be very careful not to make too many bubbles, because that will interfere with the next step, the filtration. So just take some time, squeeze this around, a couple minutes, not a hurry. We want to make sure to give the cells plenty of opportunity to be broken open. The detergent will help to break open the cell membranes, releasing the DNA into the solution, and then the detergent and the salt will help to stabilize the DNA molecules so that they can be extracted. Okay, so once this has been really well mixed and you've broken open the cell membranes from the strawberries, then what we can do is pour this mixture into the filter. Stretch a coffee filter over the top of a cup. You might want to hold on to that while you pour. Purpose here is to get all the big chunks out. The baggie can be thrown away. And then let the liquid drip. Now to help get that dripping, if you take your filter there and you can squeeze it gently to help the liquid come out, but try to make sure that the liquid is going through the filter, not around the side. So gently squeeze that until all the liquid is through, and then when you're done with that step, you can throw away the filter. Okay, in the next step, we're going to actually begin seeing the DNA. I'm going to take this bottle of ice cold alcohol, and I'm very carefully going to pour it down the side of the cup. I'm going to hold the cup here. My thumb, the bottom of my thumb, is right at the top of the liquid level. I'm going to pour this in until the liquid reaches the top level of my thumb. I'm trying not to let these mix. the ice-cold alcohol is going to form a layer on the top of this mixture, and because the alcohol does not dissolve the DNA very well, the DNA is not very soluble in the alcohol, right at the interface where the alcohol and the strawberry juice meet, that's where the DNA is going to become out of solution. It's going to begin to precipitate and rise into the clear layer. So you want to give this a little while. It takes a few minutes, and while you're waiting, you'll begin to see at the interface where the two meet some kind of goopy material form and start to rise in the solution. And you can take a stick and you can begin to mix that around and see how much you can get of that goopy DNA. That material there is the heritable material, the DNA. It doesn't dissolve very well in alcohol. It dissolves better in water. Right where the two meet, the alcohol and the water, it comes out of solution, and it's lighter than the alcohol, so it floats, and then you can extract it. See how much you can get. If you want to keep it, I'll give you a little vial that you can use. If you put a squirt of alcohol in it, you can collect some and put it in there.